So I'm so pleased to introduce our second keynote speaker of today's meeting, Dr. Roel Veerhaak. Roel is a professor and associate director of computational biology, and he's also the Florian Deschênes Rocks chair for genomics and computational biology at the Jackson Laboratory. Roel received his Ph.D. in medicine at the Erasmus University Medical Center in Rotterdam, Netherlands. Then, after a short stay as a research scientist at the laboratory of Dr. Peter Ward, Roel worked with Dr. Matthew Mason at Dana-Farber Cancer Institute and the Broad Institute of MIT and Harvard as the postdoc fellow. Roel was recruited by MD Anderson Cancer Center as a tenure-track assistant professor in 2010. So I would say during his postdoc training and early independent research, Roel established his sentinel research field in brain tumor genomics and became a world-renowned researcher bridging genomics and cancer biology seamlessly. Dr. Veerhaak has published over 140 journal papers with over 80,000 citations. Many of them are published in high-profile journal papers as the first author or senior authors, including Nature, Cell, New England Journal of Medicine, Nature Genetics, Cancer Cell, etc. So some revolutionary work includes, but not limited to, redefining the glioblastoma subtypes based on genomics data, characterization of somatic genomic landscapes of glioblastoma and many other cancer types, and the first gene fusion landscapes of different cancer types, etc. So he has received numerous rewards, such as the highly cited researcher Daniel Dan Holt Visiting Professor of Erasmus University, Angeline Early Career Professor, AAAS, Martin and Rose Watchtail Cancer Research Award, finalist of Robert Chamberlain Distinguished Mentor Award, Adult Basic Science Award, and many NIH Foundation and other foundation grant awards. So Roel has successfully trained many young scientists as well. So many of them are independent researchers right now. So just a couple of years ago, Roel gave me an exact time. So by in-depth analysis and exploration of brain tumor genomics or genomes, Roel established the field of extrachromosomal DNA amplification in cancer. So this is a new and fascinating field. His group combines wet lab and computational approaches to investigating the extrachromosomal DNAs and their functions. And they have published pioneering work in Nature Genetics, Cancer Cell, Cancer Discovery, etc. So today, Roel will share their recent work on the DNA amplification in cancer genomes. So let's give a warm welcome to Roel. Thank you.
So thank you very much, Sitchin, for that exceptionally generous introduction. I feel a lot of pressure now to give a very, very good talk after after you were listing so many accolades that came with our work. I'm excited to present here today, if only because the Informatics Institute is part of the Heersink School of Medicine, and myself being originally from the Netherlands, it makes me proud that your institute is named by and for a Dutch citizen, as a matter of fact, because Dr. Heersink was born in the Netherlands and then became a very successful entrepreneur, of course, in the United States. I'm delighted to speak to you. I see a lot of friends on the list of attendees to this talk, so thank you again for hosting me. I don't want to claim that my research and my lab's research has founded in the entire field of ecDNA, but we are enthusiastic about it, that's for sure. And I'd like to talk today about therapeutic approaches to target extrachromosomal DNA amplifications in cancer. I understand with Zoom, it's a little hard to interrupt, but if at any point there are burning questions, I'm more than happy to take questions during the talk, and we don't have to wait until the end, but I also recognize with Zoom, this is always a little bit more complicated. So either way is fine. So I'd like to make disclosures. I'm a co-founder of Boundless Bio, a company that's developing therapeutics against extrachromosomal DNA cancers. And I am a consultant for Stellanova Therapeutics. So my lab has focused on brain tumors for the last decade and a half, in particular glioblastomas, but also other types of brain tumors. Glioblastoma is a highly deadly brain cancer, and it was the first type of cancer characterized and analyzed by the Cancer Genome Atlas. And the first TCGA paper in 2008 was therefore a glioblastoma paper. And in this paper, as well as in follow-up papers, such as the 2013 TCGA paper on glioblastoma, we identified a number of key pathways that were altered in almost all glioblastomas. These key pathways included the receptor tyrosine kinase pathway, the cell cycle pathway, and the p53 pathway. If you look at the individual genes that make up these pathways, you find genes like EGFR, MDM2, CDK4, and so on and so forth. And if you then look, as is shown in these pathway plots, at the mechanism by which these genes are altered, you'll find that many abnormally activated genes in glioblastoma are genomically amplified. So instead of 2 copies of the gene, you've got multiple, sometimes dozens of copies of that gene. And in fact, GBM as a disease, and we know that in part by the many pan-cancer analyses that have been performed using TCGA and other data sets, we know that GBM is as a disease highly frequently activated by gene amplifications, more so than most other cancers. Extrachromosomal DNA, or ecDNA, and I'm going to use that term quite a lot, so ecDNA is an extrachromosomal DNA, in cancer were first described in 1965. I'm making the jump from extrachromosomal DNA, from genome and gene amplifications to extrachromosomal DNA. Because as you'll see, this is one mechanism that cancer cells use to amplify oncogenes. In 1965, in these now what we now know as landmark papers, these tiny speckles on metaphase spreads, and I believe that I should be able to find a pointer, hold on. These tiny speckles, as are shown here, could be easily mistaken for debris on the slide, but in fact, because they were frequently existing in pairs, they were recognized as actually being DNA bodies, small DNA bodies or minute DNA bodies. And because they occurred in pairs, they were labeled double minutes. And in some diseases, like neuroblastoma, which happens to be the type of cell that is shown here, the pathology reports actually detail the type of amplification that is happening in that tumor. So double minutes, or DMs, is still a term that's being used today. Although in the last few years, we've gradually moved over to using the term ecDNA instead, because it's more encompassing. Not all amplifications occur in pairs. It's just that in metaphase spreads, they always show up as such. A few years later, in a paper from Gilbert, the discovery was made that double minutes, the DNA segments that were constituting double minutes, were the same as some DNA segments making up HSRs, or homogeneously staining regions. So it was recognized that double minutes were actually a mechanism for gene amplifications, because HSRs are a mechanism for gene amplifications. And another few years later, by Fred Alt, still active at Boston Children's, showed that the sequence making up these double minutes actually contained sequence of genes in particular, and again, this is neuroblastoma cells. In this case, the gene was MYC-N or NMYC, an oncogene that's frequently amplified in neuroblastomas. It's actually a poster child disease for ecDNA, and NMYC is a poster ecDNA, if you will. So in just a few years, we understood that double minutes were a cancer cell specific entity, and that these were elements that were coding for oncogenes. However, since then, we have not really made much progress in the understanding of ecDNA. So as I mentioned, my lab studies glioblastoma and other brain tumor types. And in, I would say, 2012, we connected with Ana de Carvalho and her mentor, Tom Mickelson, both at the Henry Ford Hospital in Detroit. we developed a research project where we were going to evaluate the level of genomic drift that would occur when you create model systems from patient-derived tumors. To understand better if these patient-derived models are relevant in, for example, evaluating drug responses. So Tom and Anna collected glioblastoma tissues, 13 in total, so 13 different brain tumors or glioblastomas. Derived cultures from them in glioblastoma, those are called neurosphere cultures. So Anna established cultures from the patient tumors. And then she injected those tumors into mice as xenografts. And she did that in triplicate. And therefore, we had 13 GBMs, 13 neurospheres, and 39 xenograft models. She then collected a specimen from each of those and sent those down to Houston, where we sequenced them. And we, as Hoon Kim, who was at that point a postdoc in my lab, we then generated this landscape figure where you can see the 13 different sets of samples at the top, 13 patients. And here we are showing genes that are frequently altered in glioblastoma. And you may appreciate, if you, for example, focus on patient HF3055, that when PTEN is lost in the primary tumor, it's also lost in the model systems derived from that primary tumor. And actually, that's true for all genomic alterations, suggesting that the genomic landscape, the genomic characteristics are actually pretty stable as model systems are being established. However, we also noticed some unusual patterns. For example, here's a case of a HF2354, a focal MYC amplification. MYC, a very potent oncogene on chromosome 8. And the MYC amplification was not detected in the patient tumor. But when Anna had established the culture and when that culture was injected into mice, a focal MYC amplification consisting of 2 different DNA segments had materialized. Moreover, Hoon recognized in his analysis that there were sequence reads connecting these two DNA segments. And in fact, you could create some sort of circular path going from one segment to the other. This motivated us to ask Anna to perform FISH assays using probes against MYC and probes against chromosome 8 to determine whether the MYC gene was still residing on chromosome 8. And Anna did this in interface spreads, shown here for the tumor and the PDX, and also for the neurosphere culture, where she could establish metaphases. And especially in the metaphases, it's very clear that MYC is extrachromosomal. The red dots are all outside the blue DNA signal and outside the chromosome 8 and unrelated to the chromosome 8 signal. In other words, MYC is extrachromosomally amplified in this particular tumor. We then noticed similar patterns for many other oncogenes in most other samples. So here on top is again the MYC example, but a gene like MDM2 we also found to be extrachromosomally amplified. MDM2 is particularly interesting in a way because the gene name stands for murine double minute 2. So researchers have long suspected that MDM2 is an extrachromosomally amplified gene. Also, common receptor tyrosine kinases in GBM, such as PDGFRA and EGFR, we discovered to be extrachromosomally, not chromosomally amplified. We in total performed 65 FISH assays, and only a few are shown here. And in all 65 cases, the conclusion was that there was an extrachromosomal amplification. We also recognized the very dynamic nature of ecDNA amplifications. Here are 2 different and totally unrelated patient samples, HF3035 and HF3077, in which we detected a focal amplification of the oncogene MET, also on chromosome 7, also because EGFR is also on chromosome 7. So MET is focally amplified in these 2 patient samples. But in the neurosphere culture derived from those 2 patient samples, the 2 independent neurosphere cultures, the MET gene is actually wild type copy, as you can see here. There's no amplification detected. But when we characterized the xenograft models, we discovered that the MET amplification had returned. And it's the exact same MET amplification as you may be able to appreciate by looking at the boundaries of the focal amplification in both cases. And by the way, the boundaries differ between 3077 and 3035, confirming that these are entirely unrelated amplifications. So we have an example of a focal amplification that very rapidly disappears, although there must have been a very minor subset of cells that still contained the MET amplification. But there must have been a clonal selection process selecting against cells containing the MET amplification. But when these cells were put back into a microenvironment that could support the MET gene, it's a cell surface receptor, so there may have been a ligand interaction. When the cells were put back into a microenvironment, cells containing the MET amplification were again selected for. So you have a very rapid modulation of MET levels enabled by the fact that MET is amplified in both tumors on an ecDNA structure. From the same samples, we did long read sequencing to entirely reconstruct the amplicon. So we constructed a very circular amplicon for both tumors and observed that there's no centromeres on these residing on these structures. So one, extrachromosomal DNA is circular, and two, we see a lack of centromeres. And the absence of centromeres therefore suggests that ecDNAs segregate unevenly during mitosis. This is schematically shown here in a figure put together by Roel, who is a student at the Charité Hospital in Berlin in Germany. So we see in this schematic depiction, we have a nucleus with a single ecDNA. Upon replication of the ecDNA during mitosis and the splitting off of the two daughter cells, by random chance, one of the two daughter cells contains both of the ecDNA replicates and the other daughter cell contains none. And as this process repeats itself, after only a few cell cycles, you might have a tumor cell that's endowed with most of the ecDNAs and others with very few. And if the cell with most of the ecDNAs has some kind of competitive advantage, you now get very rapid selection for ecDNA positive cells, as well as a high degree of intratumoral heterogeneity, cells containing different loads of ecDNA. And when you do a FISH assay or a similar type of staining exercise, you will actually very quickly appreciate the high level of diversity that exists cell to cell in ecDNA burden. Now, coming back to the example I gave you with the MET amplification, as a reminder, MET was amplified in the tumor sample. It was wild type in the neurosphere, but then returned in the xenografts. When we evaluated the sets of point mutations in the same cells, again going from parental tumor to neurosphere to xenografts, we found that point mutations replicated very consistently. There was no depletion of patient mutations. All the mutations detected in the patient sample were found in the neurosphere, and all the mutations found in the neurosphere were found in the subsequent xenografts. True for both samples, maybe barring one minor clone mutation there. So what our data strongly suggested, although being indirect circumstantial evidence, is that mutations which reside on linear chromosomes segregate evenly, but focal amplifications residing on an ecDNA, such as the MET amplification here, segregate unevenly. And this work was published in 2018. What our analyses also informed us is that we could use computational analysis of whole genome sequencing data to detect circular extrachromosomal amplicons. In fact, others noticed this too. So Vineet Bhavna, a collaborator at UCSD in San Diego, developed a tool called Amplicon Architect that they published in 2019. And this tool works pretty simple. It works by identifying seed regions. Seed regions are made up of DNA segments above a certain copy number threshold of a certain size, and then mapping sequence reads onto the seed region until a circular path can be constructed. And if a circular path can be constructed, only then the conclusion will be drawn that there is a presence of a circular extrachromosomal DNA. We used the amplicon architect tool to classify first amplicons. So we classified amplicons, again defined as DNA segments of more than 4 copies and at least 10 kilobases in size. And we grouped amplicons into 4 different amplicon classes. The first class were breakage fusion bridges. This is another type of focal amplification, and it's very hard computationally to distinguish between breakage fusion bridges and circular amplicons. Therefore, we prioritized breakage fusion bridge amplifications. All amplicons that contained a signature of a breakage fusion bridge were classified as BFBs. Of the remainder of amplicons, those that had a circular path were classified as circular amplicons. We had a class of heavily rearranged where we were unable to create a full circular path, but that were amplicons that were more complex than others. And then finally, linear amplicons. Then after classifying amplicons, we classified tumor samples. And here we had the order reversed in the sense that all tumor samples containing at least 1 circular amplicon were classified as circular, then breakage fusion bridge, then heavily rearranged, then linear, and then finally tumor samples in which no amplicons were detected. And this work, this classification system was developed by, again, Hoon Kim, but also Nam Nguyen, who at that time was a postdoc in Vinit's lab. And then we applied this model, but others did so too, following our example. And in total, these various publications and other ones that are in submission have found that across primary, untreated, newly diagnosed cancers, ecDNA is still highly frequent. In a disease like glioblastoma, 60%, and we think this is a conservative 60%, of tumor samples contains at least 1 extrachromosomal DNA. This is also true for a subset of pediatric tumors like neuroblastoma in particular, and many other cancer types. And in our work, which we published in Nature Genetics in 2020, we also showed that the presence of an ecDNA, when we compared survival of cases classified as circular versus other amplicon classes, the presence of an ecDNA associated with poor patient survival. And this is not only true in a Kaplan-Meier analysis, but also in a multivariate analysis where we consider tumor type as a variable. So independent of tumor type, the presence of a circular DNA predicts poor prognosis. In 2013, Ramin Baroukhim and colleagues at Dana-Farber published one of the first pan-cancer studies looking at copy number changes across a wide variety of tumors. And they used Affymetrix SNP6 microarrays to do so. And with this now older technology, they would have been unable to distinguish between linear amplifications and circular extrachromosomal amplifications. In their landscape paper, they identified a number of genes to be highly frequently amplified across many cancers. When we performed a similar analysis using the whole genome sequencing data sets we analyzed, we found that almost all the genes shown in Ramin's 2013 paper were again found in our analysis of genes frequently amplified on circular DNAs. In other words, extrachromosomally amplified genes are the same as are a blueprint, are reflective of those that are commonly amplified. All genes that are amplified are sometimes amplified on an extrachromosomal DNA. Now, why would we actually care about extrachromosomal DNA? I mean, this is all interesting biology. I think we can all agree on this. And it's a peculiar finding at minimum. But why would we actually really care about it? So one hypothesis that we and others have is that ecDNA as a structure is a targetable entity. So instead of targeting the cargo gene, we might be able to target the circular structure. We all know about targeted inhibitors such as trastuzumab, which is used in treatment of ERBB2 amplified breast cancers, and how much they have done to improve patient outcomes. So blocking oncogenes is a way to target cancer. But many oncogenes are currently undruggable: MYC, TERT, cyclin D1, cyclin E1, and so forth. And we know that in some tumors, these genes are extrachromosomally amplified. If we can target the structure, we can target currently undruggable tumors. We hypothesized that if ecDNAs are cancer drivers, they should be maintained over time. They're not flukes, they're not events that just pop in and out. They should be maintained as a tumor progresses from a primary tumor to a liver metastasis, a lung metastasis, or a lymph node metastasis. And we're fortunate that we have available the Hartwig Medical Foundation dataset, a resource that is developed by a foundation in the Netherlands that is sequencing, whole genome sequencing advanced cancers, in particular metastatic cancers. If you regularly read the journal Science, that only in the last 5 weeks alone, you've seen 3 papers using the Hartwig dataset. It's truly a phenomenal dataset. And I would encourage anyone interested in studying cancer to try and get access to the data of the Hartwig Medical Foundation, because it's truly an unparalleled resource. We analyzed the Hartwig metastatic dataset, which again is all advanced cancers. Many are metastatic tumors, many are biopsies from lymph nodes and from liver metastases. And in doing so, we worked with, again, Hoon Kim, who is now an associate professor at the Sunken Kwan University in Seoul, Korea. And discovered at least one circular amplicon in 41% of the Hartwig metastatic tumor set. That's significantly higher than what we've observed in primary cancers. But keep in mind that the set of diseases included in Hartwig is not the same as the set of diseases characterized in the Peacock TCGA studies. But when we do a head-to-head comparison, for example, of breast cancer, we found 24% of primary untreated breast cancers to contain at least one ecDNA. But of metastatic breast cancers, this is 64%. Colorectal, 5% to 40%. Prostate, 1% to 47%. In other words, the frequency of ecDNA dramatically increases as cancer progresses. And this brings me to the first hypothesis, the first way by which we may be able to think about targeting ecDNA. Given the very consistent but substantial increase in ecDNAs between at the first diagnosis of cancer to the progressed cancers, if we are able to block the formation of ecDNA, we may be able to block the activation of oncogenes and better treat patients. Here's one example of an extrachromosomal DNA. Focal cyclin D1 amplification detected in tumor cells that were derived from the bone of a patient originally diagnosed with breast cancer. This patient was diagnosed at some point, but we do not have whole genome sequencing data. But we do know that after diagnosis, the patient received chemotherapy and hormone therapy, which is common standard of care for breast cancer. Despite these treatments, the tumor progressed and metastasized to the bone, and at that point, a biopsy was obtained. And in the biopsy, the whole genome sequencing data from that biopsy, we find a cyclin D1 amplification that is fairly complex, as you can see the variable levels of copy number, but also the many lines between different DNA segments. Following this biopsy, the patient underwent additional chemo and hormone therapy. Again, this is standard of care for this type of disease. Despite these treatments, the tumor kept progressing, and in fact, a new biopsy was obtained from a totally different site from a totally different biopsy site, namely the skin. You can maybe already appreciate that there's a high level of similarity between the amplification of cyclin D1 in time point 2 in the bone and the amplification at time point 3 taken from the skin. There's a high similarity, suggesting that in this particular tumor, the cyclin D1 amplification is a relatively clonal event. We performed a multi-time point analysis of a cohort of 156 patients. These were patients included in the Hartwig Medical Foundation set, but also from the Glioma Longitudinal Glass Consortium that my lab has compiled. In total, we had 156 patients with multiple time points and whole genome sequencing data. At time point 1, half of the cohort, 78%, contained at least one ecDNA. The majority of those ecDNAs have been maintained over time at time point 2. So we see stable propagation of ecDNA elements between time points, suggesting that they're active, they're relevant, they're drivers of the disease. And in fact, at time point 2, we see a further increase in ecDNA frequency in this cohort of mixed cancer types and mixed biopsy locations. So ecDNAs replicate. We don't know the biology of ecDNA replication is currently unknown. It's conceivable that different mechanisms exist to replicate ecDNA than for linear DNA. And if we would be able to figure out how to perturb ecDNA-specific replication machinery, this may be our second targeted ecDNA targeting opportunity. I want to briefly remind you of the fact that ecDNAs are circular and lack centromeres, as shown in our paper from 2018, a paper from Paul Michel's group, who is now at Stanford, who used confocal and light sheet microscopy to take beautiful images of the nuclei of ecDNA containing cells, showing that ecDNAs in real are actually circular. And again, because of this circularity and the lack of centromeres, it's conceivable that ecDNAs segregate unevenly. And I would argue that our 2018 publications provided some pretty strong evidence that that is indeed the case, that uneven segregation takes place. But this has never been conclusively shown to be the case. And Hee Yi, who is a postdoc in my lab, who is currently on the job market and is a highly talented, and I very strongly endorse her application to faculty positions. When she joined the lab, she was very interested in understanding extrachromosomal DNA and decided that what we really needed, what we really lacked, were tools to study ecDNA in live cells, to study the processes, the behavior of ecDNA over time. She leveraged the CRISPR technology that was available to us in our institution to develop the EC-tag system. EC-tag, in short, uses guide RNAs that bind to ecDNA-specific breakpoints. The tail of the guide contains puff binding sites that can bind puffs and can bind clovers. Clovers are fluorescent molecules. In short, we can label ecDNA-specific breakpoints. And because we can label ecDNA breakpoints using a guide RNA, we can study ecDNA over time in live cells. She then used the EC-tag signal to provide, to my knowledge, the first direct demonstration that uneven segregation truly happens. Here's a cell containing marked ecDNA. The cell is in pre-mitosis, and here the cell has gone into mitosis. As the cell is in mitosis, we think that the DNA is too condensed to maintain the guide RNA binding. So we lose our signal temporarily, but it also quickly comes back and we can keep studying the same cells. And as you can appreciate, at the end of the mitotic cycle, the two daughter cells contain a very different number of ecDNAs, showing directly for the first time uneven segregation. And he repeated this for many cells, and we published this paper just a few months ago in Cancer Discovery. And when we compare the ecDNA burden between the two daughter cells of a number of replicates, we find no correlation in the number of ecDNA copies between the two daughter cells. Whereas in contrast, and naturally, when we look at the chromosome 7 and MERK4 controls, we see a correlation 1 of 1 or close to 1 because there's even segregation. Which brings me to the third potential option for targeting ecDNA positive cells. If we would be able to figure out how to direct segregation, we may be able to direct segregation so that ecDNAs are no longer ending up in the nucleus. And if they don't end up in the nucleus, they're unlikely to be active. So this may be a third ecDNA targeting opportunity. How am I doing on time? Oh, it's 27. Okay, I've got a few minutes left. Good. Finally, using the same technology, EC-tag, he performed time-lapse imaging. And as you look at this particular nucleus, and the green marks here reflect individual ecDNA molecules, you see that these ecDNA molecules are converging to the same location in that nucleus. And that's actually non-random behavior. We repeated this experiment many times, and again we see, and again we see this convergence of ecDNA molecules, suggesting that ecDNA molecules cluster, and we do not see such behavior for our controls. And these interactions between ecDNAs are non-random. We compared ecDNA interactions and the length of time of the interaction to telomere interactions. Because of course, there's many telomeres in a given chromosome, in a given nucleus, so it's a great control. Telomere interactions are highly random, highly diffuse. ecDNA interactions are not, they're highly sustained, suggesting that ecDNA interactions and ecDNA clusters or hubs have a function. And when we imaged cells over a 48-hour period, we found evidence of ecDNA clustering in over half the cells, and we never find it, or maybe once, for our controls. So again, the clustering is a non-random behavior of ecDNA. And he then stained nuclei using ECTag, but also with RNA polymerase II. And we found that when ecDNAs clustered, there was a higher overlap with RNA pol-2, suggesting that these hubs, these clusters or hubs, are actually functional, and again, not for controls. Indeed, when we found ecDNA hub formation, we see a significantly higher level of RNA expression. So we hypothesize that ecDNA hubs are functional units of ecDNA, and that most of the transcription of the cargo oncogenes actually happen in the hubs, not so much in the single molecules. The hubs may share each other's transcriptional machinery, the enhancers residing on their structures to activate transcription of cargo genes. And this brings me then to my fourth opportunity for ecDNA targeting. If we can perturb the hubs, we may be able to inhibit ecDNA function by limiting transcription of the oncogene. So I'd like to summarize. Extrachromosomal DNA elements are vehicles for oncogene amplification that are frequent in cancer. We find ecDNA frequency to be highly increased in aggressive cancers, glioblastomas and sarcomas, but also metastatic advanced cancers. And we think there's multiple opportunities for ecDNA targeting that may be able to provide a therapeutic benefit to cancer patients in the future. So with that, I'll end. I'd like to thank my lab, an extremely enthusiastic and productive group of individuals, as well as the many collaborators that have worked with us on this. Hoon Kim, my former trainee who is now at Sunkyun Kwan, Chailin, a close colleague here at JAX, Vineet, Paul, Albert Cheng at Arizona State, Anton Hansen at Gaite, and many others. And thank you for your attention, and happy to take questions.
Thank you, Roel. This is fantastic, and give a longitude of view of exome DNA and also recent focus. That's fantastic. So any questions from the audience? Feel free to unmute yourself to speak up. Irvin, probably I saw your question. If you can. So I will repeat probably. Very impressive and very nice presentation. Do you see ecDNA as a tumor-specific event only, or do you think they also have a physiology function?
Hi, Erwin. It's nice to see you here. Hi, Erwin. This is a great question. So we see circular DNA elements in many cell types. In fact, you have one of the world's experts on this at your institution in the form of Dr. Dada, who's also here at this meeting. My lab, I should clarify that my lab so far has only studied ecDNAs in cancer cells. But this other type of ecDNA, these other type of circular DNAs are different from the ones we detect in cancer cells because they're much smaller, typically less than a kilobase. Whereas the ones we see in cancer cells are typically 50 kilobase or much longer than that. Also in cancer cells, ecDNAs are a mechanism for gene amplification. Whereas in normal cells, you don't want gene amplification in most cases because it would cause cancer or dysplasia or something like that. So there are multiple types of circular DNAs, multiple type of ecDNAs, but they're very different between normal cells and neoplastic cells.
Thank you.
Roel, this is an India. How are you doing? Great talk. This is the first time I actually heard the talk in a historical context. So it was really, really impressive. So I have a couple of questions. So you have characterized so many of these circles now in cancers. Do you see examples where they don't carry any obvious oncogenes?
Yeah, hi Ninja, nice to see you. So yes, absolutely. It's hard to put an exact number on this, but I would say if I had to put a number, let's say 10% of ecDNAs, or maybe 5%, or some smaller percentage, do not appear to contain genes. But they then contain regulatory elements, in particular enhancers. So we see enhancers, we see an enhancer-only ecDNAs.
But I was wondering, when they do contain genes, do they always have oncogenes, or are there examples? Could this be a gene discovery way, sort of the way MDM2 was discovered as a gene carried on these circles?
Yeah, so the majority contains oncogenes, but the term oncogene, there are lists you can compile from literature as this is a set of oncogenes, and other genes are not oncogenes. But as you understand, there's a twilight between what we call oncogenes. At some point, we did compile a list of 24 oncogenes, and that we use as our standard set, and we can debate at length of whether our list of 24 is the right set or the wrong set. We appear to see functional differences between ecDNAs containing any of those 24 oncogenes and ecDNAs containing other genes, and that they typically are amplified at higher levels. So our oncogene list ecDNAs appear to have more copies than our non-oncogene ecDNAs. And I'm sure if we go into more detail, we'll find more differences.
And the last question is actually related to Yoonhee Yee's work. By the way, you should know that she is coming back for an on-site visit when she returns from Korea. So the question is, do you guys see any evidence of integration into the genome with those visualized circles?
Yeah, it's another great question. And as you know, and I've talked about, I highly strongly endorse Inhee's application at UAB. And I'm delighted to hear that she's invited to return. I suspect that in order to see reintegration of ecDNAs in the genome, we would have to follow them for a longer time. And we have not seen it happen yet. But from other papers and other work, we know that reintegration is likely to happen. It's a confusing process to me in the sense that if there is reintegration, the only way we are able to detect it in sequencing is if there are enough cells containing the exact same integration, which means that there has to have happened some kind of clonal selection. So I'm still highly confused by reintegration, but especially from neuroblastomas, we know reintegration is often happening because we see NMIC being found at other locations than chromosome 3, which is NMIC's natural habitat. So we see NMIC in chromosome 17, NMIC in chromosome 8, and so on and so forth. So we know reintegration happens, but I'm highly confused by it, that's for sure.
Thank you.
Some type of question, probably you can unmute yourself to ask.
I have one additional question. So, Roel, in the beginning, you showed some examples of, maybe it was glioblastomas who initially had some form of amplification, and then when they were grown in as neurospheres, they did not evidence those anymore, and then they would reappear, in the mice. And it had been known for a long time that indeed in cell culture, at least for glioblastomas, you would not see those amplifications anymore with double minutes and so on. So do you think this happens because in cell culture, they simply have a disadvantage in fitness, or because they are not needed because, I don't know, there is so much stimulation already through the medium that maybe there is a disadvantage for, energy for the cell to maintain them? What's your view on that?
So we have cell cultures with many passages that have maintained their ecDNAs. So I don't think there's a one-size-fits-all answer. We've seen papers in literature that have suggested that ecDNAs, in one way to view ecDNAs, are a mechanism of tumors to handle stress coming from limited nutrients, limited oxygen, and so on and so forth. In cell culture, there's not as much stress, so perhaps that is one explanation why there are mentions of losing ecDNAs in cell culture.
Sam, probably you can ask quickly, and Lung has a question too, right? Well,
sure thing. Yeah, thanks so much for your talk, Dr. Veerhaak. Really enjoyed it. In one of these talks, she had mentioned a relationship between Epstein-Barr virus and extrachromosomal DNAs, and it really struck me. I'm a trainee in an oncolytic herpes simplex virus, which isn't the same virus, obviously, but it's a herpes viridae family virus. And one thing that we always are interested in kind of describing is how oncolytic HSV kills multiple subtypes of tumors, how it's not like a single target treatment like a checkpoint inhibitor, for example. And I was just curious if, since you said that the ecDNAs are present in broad subtypes of tumors, if you're familiar with any relationships between extracellular DNAs and viral interactions.
Yeah, there's really nice work from Vineet Bafna's lab, collaborating with others, showing fusions of extrachromosomal DNA and viral sequences, particularly in esophageal cancers and gastric cancers driven by EBV. I think there's a lot to be learned there. There's a lot to be discovered there. I think the whole area of ecDNA biology, we know so very little. And this is one of the many areas where I think we would be able to make a lot of progress by just studying how that happens, why it happens, and so forth.
Awesome. Lung, you have a question? Probably the last one.
Yeah, so it's always very inspiring to hear your talk, Roel. So you just mentioned that this kind of so-called heterogeneity of this ecDNA. Do you think that it is possible to characterize this ecDNA at the single cell level?
Yeah, absolutely. I mean, we and others actually in India had a very nice paper not too long ago using ATTIS to detect or to profile ecDNAs. And we and others are using those methods to detect ecDNA in single cells. It's far from perfect because you don't get broad coverage of the whole genome, obviously, with a single cell ATTIS. But we think we can, with some degree of uncertainty, separate ecDNA containing and ecDNA negative cells.
Okay. Thank you.
Okay, thank you. Thank you, Roel. This is fantastic. So we are just running, we are just hitting the time. So thank you again. Thanks for the great talk, and thanks for the great questions too.